Dear students, today I'm going to discuss about the topic hepatitis C virus. It is there in your course MMB205 module 3. In module 2 we have already discussed about hepatitis A and B virus. Today we are going to discuss about hepatitis C virus. This particular virus, the study of this particular virus is very important because this particular virus is responsible for the chronic hepatitis, or that is actually creating hepatocellular carcinoma. Now I'm directly entering into the main topic. Hepatitis virus, hepatitis C virus in fact, was first identified in 1989, but the first blood test became available in 1992. This particular virus was first, earlier it was known as a non-A, non-B hepatitis, means NANBH, to differentiate the hepatic disease caused by the viral origin. It is a non-alcoholic hepatitis. This particular virus spread through blood-to-blood contact, means parenteral route. And hepatitis C virus, henceforth I will just explain it at HCV. So this particular virus falls in the family Flaviviridae and genus Hepacivirus. Most patients infected with HCV have chronic liver disease. And this chronic liver disease, which progress to first fibrosis, then cirrhosis, and then hepatocellular carcinoma, that is called HCC. And the most alarming fact is that to date, no vaccine is available to prevent hepatitis C infection. What is the morphology of this particular type of virus? This particular virus, along with hepatitis B, like Den particle, they are also spherical in shape, they are enveloped. It's a single-stranded RNA virus, and the diameter is around 55 nanometer, where in case of Den particle, we have seen around 42 to 47 nanometers. So they are a little bit of larger size. The genome is at approximately 9,600 bases that encodes 10 structural and regulatory proteins. I'm going to discuss this. What are the 10 structural and regulatory proteins? So particularly the envelope protein, there are two different types of envelope protein. They are called the core protein. And these two proteins are called E1 and E2. These are two glycoprotein. These two envelope protein undergo variation during infection due to hypervariable regions within their gene. And these viruses are sensitive to ether and sensitive to acid. So this is the structure of hepatitis C virus. Here you can see here. This is the glycoprotein. This blue color is the glycoprotein. This is called the enveloped glycoprotein E2, and this one is this green one. This is the E1 enveloped glycoprotein. They contains this fatty envelope, and this is the cell of protein, and this is the fatty envelope. And here this is a hexagonal symmetry of the capsid, and within that they have a single-stranded RNA. So this is their nucleocapsid. So now we are going to discuss about their genome. So as I said that this particular virus contains 9,600 nucleotide base and encodes 10 structural and non-structural proteins. So what are the structural and non-structural proteins? Here you can see these are the structural and non-structural proteins, say P22, GP35, GP70, P7, P23, P70, P8, P27, P56 or P58, and P68. And these are the genes. So these are the genes. They are actually they are synthesizing or encoding some proteins. Each and every gene, or maybe multiple genes, they are responsible for the synthesis of different type of proteins like nucleocapsid, envelope glycoprotein. These are the spikes E1 and E2. And here this is transmembrane protein, here the anchorage protein, then proteases, RNA helicase, cofactors, RNA polymerase, and so on and so forth. Here you can see these are the genes. They are actually synthesizing. This is the gene. These are the genes P56 and P58 and S5A. They are the genes actually encoding a resistance protein, and that particular protein is resisting interferons. That is why this particular type of virus, HCV, has the ability to escape immune response of the host cell. So that is why they are more dangerous. This is their genetic types. So HCV has 6 distinct major genotypes, and here you can see HCV, HCV 1A, 1B, 1C, then 2A, 2B, 2C, 3A, 3B, 4, 5, and 6. So these are 1, 2, 3, 4, 5, 6. These are the 6 distinct major genotypes. And here also in A, in B, and C, there are sub-genotypes. So the question is, why should we go for the study of HCV genetic types? Because they have some clinical significance. These genotypes or the subtypes served as a strong prognostic marker, very strong prognostic marker for HCV antiviral treatment or development of antiviral drug against HCV. Now the question is how they transmit. Particularly, they transmit through blood-to-blood contact. In what are the routes? Means recipient of contaminated blood transfusions, blood products or organ transplantation, injections given with a contaminated syringe or needle, drug abuse, and then mother to child. These are the routes. Hepatitis C virus can also be transmitted through sex with an infected person or sharing a personal items contaminated with infectious blood. So it's a blood-to-blood contact. But point to be noted that hepatitis C is not spread through breastfeeding or breast milk, food or water, or by casual contact such as hugging, kissing, sharing foods or drinks with an infected person. And also it cannot be transmitted by air, droplets, vector, or from animals. So only blood-to-blood contact they can be transmitted. Now, how they replicate? Mostly HCV replicates in hepatocytes of the liver. So the virus may also replicate in PBMCs, means peripheral blood mononuclear cells. They does not replicate via a DNA intermediate, means they will not integrate into the host cell genome like HBV, means they are not forming any proviral DNA. So they are entering into the host by these two glycoproteins, E2 and E1. They recognize a surface protein, the surface receptor that is known as CD81 present at the surface of the hepatocytes and PBMCs or lymphocytes. So CD81 will receive this and they will enter via receptor mediated endocytosis. When they made an entry into the cytoplasm through endosome or via the formation of endosome, then after uncoating there, the mRNA undergoes translation directly and they will synthesize polyproteins. Then HCV RNA replicate to form new viral RNA, and along with the polyproteins they assemble and come out from the body. or they disseminate and complete the new replication cycle. So here is this picture. You can see this picture will tell you how they replicate. Here you can see these are the two receptors. One is your CD81 receptor, another one is B1 receptor. So this E1 and E2 glycoproteins, they will interact with these two receptors, and there will be internalization via receptor mediated endocytosis. You can see here the endosome has been formed. Now they are uncoating. RNA is coming out from the nucleocapsid. This blue color is the nucleocapsid. So from there, here is the RNA, single-stranded RNA. It is coming out, and then directly going for translation and for polyprotein processing. And after the synthesis of polyprotein, they will go for RNA replication, and then they will miss for RNA synthesis, and then again they will go for assembly along with the polyproteins, and then they will come out. So this is how they replicate. Now, what is the infection? So exposure of HCV to the human. So 60 to 85% human having HCV infection for more than 6 months, they are having chronic infection. And person with acute phase or exposure of acute phase, 15 to 40% infected person can heal automatically with their immunity. So these 60 to 85% of infected person, they have the chronic disease, chronic liver disease. So 20% of these will develop cirrhosis via fibrosis. And out of this 20% of cirrhosis present, 4% per year will go for or will develop HCC, and 6% will develop end-stage liver disease. Both the types can go for liver transplant, means hepatocellular carcinoma and end-stage liver disease. Both the type can go for liver transplant or else death. So here death percentage is nearly 3 to 4% per year. But this is a prolonged way. Here you can see the arrow. This arrow signifies it took here about 30 years. It's not that so fast the disease is progressing. It's very slow. So 25 to 30 years it took to develop HCC, eventually for death. So the question will come, what happens to people with hepatitis C virus? So a statistic shows that 100 people or individual infected with acute hepatitis C, 15 out of 100 will develop no chronic disease. So 85 out of 100 can develop or may develop chronic disease. Out of this 85%, only 17 will develop cirrhosis and 2 may develop liver cancer or hepatocellular carcinoma. So this is the statistics. Now what about the incubation period? So incubation period for hepatitis C is 2 weeks to 6 months. What are the symptoms? The symptoms are mostly they are asymptomatic. But in case of the symptomatic individuals, may exhibit fever, fatigue, decreased appetite, nausea, vomiting, abdominal pain, dark urine, then gray-colored faces, joint pain, and jaundice. So 75 to 85% of newly infected persons develop chronic liver disease. And 5 to 20% develop cirrhosis, and 1 to 5% die from cirrhosis or liver cancer, HCC. So here is the developmental stage. From the healthy liver, here you can see, from the healthy liver to fibrotic liver, means inflammation continues. Inflammation has started here. And here you can see this is a cirrhosis, liver cirrhosis. And disease means here you can see blocking the flow of blood through the liver. And here you can see formation of malignant tumors. This is actually HCC, or hepatocellular carcinoma. So here's how hepatocellular carcinoma can be developed by HBV or HPV. There's a two type of mode. One is a indirect mode. Another, this is the indirect mode, and this is the direct mode. In case of indirect mode, here there is DNA damage, increment of DNA damage, mitochondrial dysfunction, and ear stress development. In case of direct oncogenicity, you can see this cancer stem cell development via DNA integration or via the core protein development. So what is actually HCV core protein? Here you can see that the core protein is actually doing all the work. This core protein, they are responsible for these blue boxes. You can see the blue boxes indicate the key driving forces for carcinogenesis. Here this means increment of genomic instability, ROS generation, reactive oxygen species generation via mitochondria mediated pathway, then transcription and proliferation, differentiation, decrement and proliferation enhancement, then vascular endothelial growth factor enhancement, and decrement in apoptosis or natural cell death. So this HCV core protein, they are actually responsible for HCC development by the following driving forces. So what is the diagnosis? So mostly diagnosis is of acute infection is often missed because a majority of infected people have no symptoms. They're asymptomatic. But if the symptoms is there, so you can go with HCV recombinant immunoblot assay, which is called RIBA, and HCV RNA testing. These are the two established methods that can confirm HCV infection is there or not. Means we can go for diagnosis with these two methods. Early diagnosis has a potential to prevent health problem and eventually development of cirrhosis or HCC. But the main thing is you can prevent, or if you have identified that, okay, this person have developed HCC, have infection of HCV, not HCC, have developed HCV, then you can go for the followings. So people who have received blood products, organs before screening for hepatitis C virus, then current or former injecting drug users, as well as the individuals of long-term hemodialysis, healthcare workers, individuals having HIV infection, individual with liver disease, infant born with infected mother. So you can go for early diagnosis. And for that, you have to follow the mentioned two method or mentioned two process. What is the treatment? We're sorry to say that this particular hepatitis, no vaccination, no such drug is there. So multiple therapy have developed. So two new therapeutic agents. One is telaprevir and the second one is bezaprevir have been used in certain countries. And here another one is ribavirin. This is the new drug that is actually using as an immunomodulator. And this is people are using, or the doctors are suggesting to use this particular drug, antiviral drug ribavirin, and this has a proven effect on HCV. So this particular drug is actually functioning as a nucleoside analog, and they are blocking the IMPs. Here you can see they are blocking the IMP. Here you can see this is nothing but inosine monophosphate, and here it is inosine monophosphate dehydrogenase. This is an enzyme, and this enzyme is responsible for the conversion of GTP. And when GTP is not formed, so the HCV RNA, they will not get nucleotide and they will not go for further replication. So what is actually happening here? So ribavirin either just favoring via immunomodulation the T helper cells 2 over T helper 1 cells and enhance host immunity to the virus, or else it will enter into the cell and then utilizing thymidine kinase of HCV and then utilizing the host cell kinases, they will convert into ribavirin monophosphate, then diphosphate and triphosphate. This ribavirin triphosphate will have the competitive inhibition with GTP. So GTP is a nucleotide, and this will nucleotide analog. So this will bind with the RNA polymerase, and then as because they don't have any free weight group at the 3' end, so the chain will not develop. This HCV RNA will not go for further replication. Here the replication will be stopped and no virus particle will be formed. So that is how you can means scientists are using ribavirin as a drug in both the ways. Here using just immunomodulation of Th2 to boost the host immunity against the virus, and here by using as an analog to block the function of RNA polymerase, or to block or to terminate chain elongation or replication of HCV or hepatitis C virus. Now, these are the target sites for chronic HCV, and here multiple drugs along with interferons are used. And these are the 9, this blue one, you can see this blue one. Here you can see that these are this is where you can block. Here first you can block the interaction between E1, E2 with CD receptors, and then you can block the entry, then you can block uncoating, you can block RNA or formation or replication, you can block assembly, you can block release also. So these are the 10 target site scientists have identified to develop antiviral drug against chronic hepatitis C virus infection. Now, prevention. So there is no vaccination for hepatitis C. So the risk of infection can be reduced only by avoiding unnecessary and unsafe injections, unsafe blood products, unsafe surf waste collection and disposal, unsafe sex, and use of illicit drugs and sharing of injection equipment. Thank you.